Okay, you're good.
Hello everyone. Good evening or good morning wherever you are. Uh, it's my pleasure to introduce Dr. Bai. So I will first start with his education. Dr. Bai had his PhD in plant pathology at Purdue University between 1991 and 1995. And he had a MSc in plant genetics and breeding in Nanjing University in China, and he had a BSc in agronomy between 1978 to 1981. So Dr. Bayer over the years had made groundbreaking contributions to the SCAB initiative and fusarium head blight resistance research, including developing the first robust FHB severity protocol in the greenhouse and identifying deoxynivalenol, which is an important mycotoxin when it comes to the end user virulent factor. And he cloned the FHB1, FHB7, FHB9, and transferring these genes into elite wheat lines or germplasms that directly had influence on resistant wheat cultivars. Dr. Bai impacted has extended globally over the years through the development of widely used DNA markers, and he has co-released 55 wheat cultivars over the years, and also pioneering genomic tools, over 350 high-impact publications over the years. And he has more training and had trained over 220 scientists over the course of his work. And he recently retired at the USDA in Manhattan, ARS. So with that, let's welcome Dr. Bai. And Dr. Bai can take the presentation on. Thank you.
Okay. Thanks, Lawrence, for inviting me to come here and for the long introduction. Actually, I work in the USDA the genotyping lab in last like about 23 years. I just retired early this year. So today, because my work actually involves genotyping of breeding material for hard and winter wheat for the egg breeding program, and also work on research on several different kind of trait of wheat, like including like rust, soluble mosaic virus, and BOIDV, and aluminum tolerance, and several other. So, but today I'm only going to talk about wheat scab resistance, because there's a scab initiative. So actually, in last 20-some years since the SCAB initiative started, I get funding for them from SCAB initiative. I really appreciate the opportunity and the support for my research. So before I started, wait a second. I'm going to give really some kind of block my screen. I give a little bit introduction on the history of scab research. Actually, with scab research started about the 1980s. So first reported in England about 1984. So then in US, in Ohio State, they reported by 1889. So that's the name of scab come from. They first reported with scab. So actually after there about in 1930 or 40, and the US have the scab again. So there are several groups started scab research, mainly focus on find the resistance. They are actually find like two types, one type 1 and the type 2. Right now we still work on it. So right now, most people work on just focus on the type 2 resistance. Actually, let's start from that time. So also some people, pathologists, just start work on the fusion race, try to classify different races. But finally, people find that not really the fusion race exist. So in 1950, in Japan, they have reported some variety have resistance, especially like one name called Shichanaga. So that one, they use classic genetics. They report about 3 genes control the resistance in that variety. So they actually use that material as a resistance source, develop a lot of variety in Japan, in the like Noring system. There a lot of variety have a resistant to a scab. So, but in 1970 to 1980, actually in China, in the Yangtze River area, so that scab occur very frequently. Almost every other year have a scab epidemics in that region. So then in China, they organize an organization they call the Chinese National Wheat FHB Collaborative Group. They're pretty actually nationwide. So they do the test on the scab material, wheat material, whatever they can find, use a method, whatever they have to test the scab resistance. Finally, they compile one book that list all the whatever material they have resistant. Then they finally they found that like Sumai-3, Wangxi-Bei, and some Chinese landrace show very good resistance. Also, sometimes they develop the scale screening method. So then they use invent a method called 0 to 4 scale. So that one used for the germplasm screening. So that scale basically when disease not spread, they like as just no infection 0, and no spread like 1, a little bit spread 2, like spread like half of that, 3, and all the 4. They kind of very kind of roughly score that disease. But that one is difficult to be used in genetic study. So it's not really like quantitative. So then in that time, in 1980, in Canada, so they are working on the D1. They found that the plant also have like D1 resistant there, so that they proposed a certain type resistant. So in actually in US in 1980, the disease epidemic occur in several states, become kind of a problem there. People realize scab is kind of something people should work on. So, but at that time, people actually stopped working on that anymore. So that time, people like even don't know how to score the FFP resistant in genetic research. And some people try to do the like a ceiling test, try to use the ceiling resistance to replace like FHB. So then they find out the ceiling resistance didn't work very well. So not correlated with fusion head blight. So also people don't have any method to evaluate the wheat scab resistance in the greenhouse. So another problem because people know nothing about genetics. So you don't know is that a minor gene or major gene or both of gene control the scab resistance. Okay. I couldn't turn off that. So then, actually, as I said, I started FFB research almost about like 40 years now. So today I'm going to give a very brief introduction of what the kind of research history what I had. So, sorry, that's my computer didn't work very well. Yeah. Actually, I started my scale research started from 1982 when I was a master student. That time, I mainly worked on the classic genetics of with resistant to scab. So in that work, mostly I have considered two things. One, based on whatever data we got, we found that the resistant to scab have major gene because when we look at disease frequency distribution, we find that it's not like a normal distribution, but we find that they have like two peaks. So based on that, we use like kind of classic genetic research method. We find that at least have this one or several, one, two, three different gene that we can quantify the effect. Also, I kind of use like a percentage of scabies spike rate, try to quantify disease resistance, use that data for the genetic study. So that work published in 1989. So then actually I moved to US in about December of 1989. So then I worked with Dr. Schenner in Purdue University. So continue work on the scab. So then we got the invitation to write a paper, a feature article on the plant disease. So this is a paper, the picture we have in that paper. But that paper actually helped a lot of people to learn how to start scab research. We report kind of a disease evaluation method, the inoculation method, and scoring method, etc. So, so then in about 1999, so we mapped like first major criteria for FHB resistant. Actually, that paper published in the Phytopathology, that in April, but in May, another paper from Dr. Anderson group reported at the same times. But at that time, we used FRP, didn't show the chromosome location, but Dr. Anderson paper that use alpha-P, they are clearly show that criteria on the 3PS. Later, people named that as FHB1. So in about 2 years later, we published another paper on the micropathology that established D1 as a variant fact by kind of showing like fusarium, when knock out D1 produced gene, that cannot cause disease spread in the spike, but only cause the initial infection. So, but then people just use that as a further evidence of D1 as kind of a variance fact. So in 2006, when we use like Chinese Spring genetic stock, like with that one arm deletion line, so we found that some line with one arm deleted become kind of resistance. We propose that with resistance scab, we have like a susceptible effect to control the scab response. So then in about in last 10 years, especially like we cloned the FHB1 in actually is 2016. Then we submit paper until like 2019 they have been published. So, but before that, the cloning gene work have been published. We developed like a diagnostic marker for the FHB1. So we just released earlier. So in 2018, for breeder to use that as a breeding tool to select for the FHB1. So the marker right now is still a major marker people use for the selecting the FHB1 in the FHB resistant breeding. Then in 2022, we developed a marker for FHB7. And in last year, we named the one gene called FHB9 in the one Chinese source. So because FHB1, FHB7, and FHB9, they have very good marker kind of diagnostic for those three genes. So in last several years, we started pyramiding the gene the 3 gene into the hard winter wheat. Actually, right now we have already get those motor material we sent out more recently to the breeder with the 3 gene pyrimidine is marker sequence selection. So those are kind of major milestone we had for the with scab research in last about 40 years. So most of my research actually focus on several different questions. One is in the beginning that we try to figure out is a major gene or minor gene, or is a gene dominant or additive? That's kind of very basic question people need to ask before they use those genes. So also plant response to the gene is that the gene just due to the resistance or lack of function of the susceptibility gene. So also what the DON function is on the resistance. So first I'm going to give some example of to show that is that the gene or dominant or recessive or additive. So in the early paper, we show that the disease distribution, like these 2 parents, P1 and P2, but F1 usually in the middle between the P1 and P2. So that means maybe like partially dominance or recessive or just kind of additive. So because depending on what the environment factor is. But for F1, that distribution pretty wide, actually just like F2. So the line here, they show the F1 distribution, because that's usually like for some gene only have a single copy, like very kind of easily affected by environment. So it kind of cross the old range. So, but for the recombinant immune line, they usually have like a 2 peak. So that actually is kind of evidence based on the classic genetic. And so that show like only like maybe 1 or 2 major gene control the disease resistance. So for the highly resistant genotype, we can see several situations. Mostly they have like 1 major QTL, like this one have FHB1 there, but also you would be able to detect several minor QTL, but those QTL may not be stable from different kind of repeats. Some you show up, some you cannot detect it. So that's one situation. Another situation, you hardly find any kind of really big QTL, but this one, further one, like this is one variety we called Hay and Zong. So this one have very high resistance, but we hardly detect a really high large QTL. But this QTL, further one like on 5AS, they show that pretty good consistency between different years. So and it's kind of not a very large effect, but it's kind of between the major and minor gene. So this one, but all other genes only show up in some year, but not in other year. Some year that couldn't detect it. So I'll show the kind of a minor effect. But one thing we notice that in susceptible parents, Wheaton, problem some of you know, Wheaton is very highly susceptible. But when you do the QTL mapping, you still can detect some QTL in some year, but even not very stable, but still some minor QTL that you can detect it. So that actually here show that multiple minor QTL additive effect. So susceptible parents can contribute to resistance. And also for the minor QTL, usually repeatability pretty poor, so probably need like a multiple year location you can detect the effect. So this just show like when 3 gene with further 3 have a little bit larger effect, we just use that 3. So when 3 genes together, they show very low percentage of disease speculate. But when none of that have resistant allele, they show very high. So they're kind of additive. So also, we found that the major QTL usually expression kind of affected by minor QTL in the background. So here when we transfer FHB1 use a marker to a different background, we use 3 different backgrounds. One is the overland, that's have a moderate resistance, and Jagger moderate susceptible, and overly highly susceptible. What we did, we used like select a 300 plant line. So then we use a marker just select for FHB1. So then the selected line we tested. So for the scab resistant phenotype in the greenhouse, if those plants have less than 50% PSS, we just kept it. So then we do the phenotyping again. Then select plant, we found that like overland have more highly resistant if we are just classify as a highly resistant, moderate resistant, and moderate susceptible and susceptible. So then we found overland have more plant have a higher resistance, but only a few plants have a moderate resistance. But for the Jagger on the middle, so that only have a few plants show higher resistance. But most plants have more plants show the moderate resistance. But in overland background, only have few plants, much fewer than in Jagger background, have a moderate resistance. That means genetic background significantly affect the like gene expression, major gene expression. So just a short summary. So for both major and minor QTL can confer resistance. It depends on what kind of accession you are started with. So actually highly resistant genotype can be due to like a major gene plus a minor gene, but also it could be like many like minor gene, minor QTL controlled resistance. So sometimes you will see, a lot of time you will see the susceptible genotype also contribute minor QTL. So QTL mainly show additive effect, but minor QTL may not be significant in many environment or background. So number of effect of susceptible QTL also affect by the expression of also affect the expression of resistant QTL. So is a resistant regulated by a resistant gene or just the loss function of a susceptible gene? So we here some evidence that the resistant controlled by resistant gene. Like one evidence because FHB7 have been cloned, we did the transgenic with FHB7. We just put like FHB7 into fielder. Fielder is kind of moderate susceptible variety. So we check the gene expression, like those are transgenic plant, those also transgenic plant. So one line they show low expression, but all others show very high expression of GST that also called FHB7. So, but the phenotype, so this highly expressed plant that show very low disease level, but this is the parents. But those one, this is a negative transgenic plant. This one low expressed transgenic plant all show higher disease level. So that means FHB7 is a kind of resistant gene, but the higher expression of this gene reduce the disease level. So this is planting in greenhouse about the 20 days after inoculation. So the right side shows the transgenic plant with a highly expressed FHB7. The left side, that's kind of also transgenic plant, but without the gene or have low expression of the gene. So this single plant, that's a picture that actually show no expression, show not the disease spreading the spike. So it kind of a surprise. So this in field, the two neogenic line of FHB7, this is a resistant one, this is a susceptible one. So another evidence is a deletion of a chromosome arm increase FHB resistant in Chinese spring. So you have like about 42 different kind of deletion line from Chinese Spring. We found that like this Chinese Spring disease level, but some other line with deleted like the 1AL, 1DS, 2AL, 3DS, 1DL. So those ones show significantly higher disease than Chinese Spring. So those ones, so we assume this because of a resistant gene on those specific arm. When this arm missing, so that the gene is gone, so the disease level increase. So this is another evidence of a resistant gene. We also did one phenotype for chrono. That's I think this is Durham. So the parents that have like chrono, they have like about a very low, relatively low disease level, but some mutant we find on the gene, one candidate gene on the 4BS, they show after the mutation, so the disease level much higher than original parent of corona. So those are the evidence of resistant gene, but we also found some evidence of susceptible gene. So that's in same experiment with Chinese Spring, like a Chinese Spring that they have like a very high, relative high disease level, but when some arm deleted, so the disease level become lower. So those arms like 4DS, 7BS, 3BL, like 7AS, the disease level become lower. So those are all kind of evidence to show probably susceptible gene on those arms of deleted the gene, susceptible gene is gone, improve resistance. Also, we did some work use kind of EMS induced Jagger mutant. We we screened that for the population for the FHB, but we found that about 3 M4 line, they like Jagger have like about 80% disease, but there are 3 of M4 line only have disease of 20 or 30%. So there's a significant reduced disease level. So then we develop a recombinant immune line. So then we mapped several minor QTL for FHB resistant from the mutant. So another kind of major evidence is knockout HRC, also that is the FHB1, can increase resistance. This one is a gene edited plant that Bob White, that's a negative plant without editing. After editing, so the plant disease level significant drop. So that means knockout of this HRC increased disease resistance. So the third question is how DON affected disease resistance? Because when I work on in the mycotoxin lab in the period, so that time we found that so when fusarium DON producing gene knock out, so those two lines show that when DON knockout, when we inoculate that with a knockout the fusarium, we found that this is a needle inoculation. Disease have no increase. That means disease not spread from inoculate site to other spikelate. But then when we spray the spores on all the head, we found that there's a little bit increase because infection site increase, but still pretty low relatively to the wild type. So wild type after like 9 or 12 days, so all spikelates get killed, like the 100% disease level. So that means just shows evidence of, as the title said, so D1 produce fusarium just cause initial infection, but do not cause disease spread in the with spike. So then people cite this as the first evidence to show the D1 as kind of variant fact. So then another example of D1 as a kind of some kind of effect resistance is FHB7. So in this paper, they show evidence that FHB7 can detoxify D1. So actually, we did an experiment on the D1 production for the FHB7 transgenic line. I said that some highly expressed line show much lower D1 production. So several other factors may also affect the FHB resistance. Probably I see a lot of reports from literature that plant height, such as IHT1, IHT2, ITA. So we mapped the QTL in the IHT1, IHTA region. So right now, the candidate gene have been identified. Right now, kind of validate the function of that, just to see if that is the gene per se affect the resistance or some other gene nearby affect disease resistance. We try to figure out that relationship with the plant height and the scab resistance. So also probably heading there, such as the PPD1 region that also affect the resistance. And some people report answer intrusion. And also another thing is groom hardness. So actually, especially like 5AL, the gene have a very large effect gene there. But the problem is when we transfer that gene, sometimes we kind of get like a hard groom, especially hard to get seeds separate from a groom. So that could be like a hard groom could be some kind of physically resistant scab infection. So actually, a little bit summary here. Resistance could be due to like a resistant QTL or loss function of susceptibility QTL. So actually, also low DNA accumulation could affect the resistance. QTL also could be associated with the gene for the morphological trait. So now I'm going to talk a little bit about FHB1, because so far FHB1 is the first reported QTL for FHB resistant in wheat. So it shows kind of relatively stable major effect, even jump up and down, but that shows the major effect in multiple background and environment. So far, still a kind of a best gene found in wheat. So, but that actually is pretty early identified from SUME3. That is in 1970. So far, I have read about more than 50 years. So, and used in breeding program right now worldwide. So this actually FHB1 is not like something magical, kind of a complete resistant to the FHB, but it only shows like partial resistant depending on what kind of a background. Some, as I say, some can be high resistant if other monoQTL help. So if no monoQTL, if that's like a susceptible gene there, they will probably, some more susceptible gene, they probably show like a moderate resistant or sometimes even show moderate susceptibility. So, but the good thing is this gene show like additive effect with most of other like native QTL. So, but in US right now, in like soft winter wheat and hot spring wheat, so they all have this lot of variety right now. But in hot winter wheat, officially only have one variety more recently released from Oklahoma. That's come from like a FHB1 backcross project. We supported from Scab Initiative, so that finally we have one variety released from that work. So have FHB1. So FHB1 actually the causal gene we identified that it's called HRC, the histidine-rich calcium binding protein. they encode that protein. So actually, when we clone the cDNA, we find that in susceptible line, neogenic line, we find kind of I have like have three exon, but they only show one band. But in resistant line, we found like 2 bands there. After we sequence, we found like at least 4 different products. One only have 2 exon. The other 3 have 3 exon, but with different. The third exon have different lengths. So actually, they have multiple product. But so when we try to do the transgenic, use resistant HRC from resistant line, but the transgenic plant didn't increase any resistance, the same as Bob White. So it didn't show any kind of effect. So from a resistant line. So, but when we knock out the allele from susceptible line, we show a significant decrease in disease level. So that means knock out the loss function of susceptible allele, the increased resistance. So, but then we did some work on try to determine which part of gene deletion will cause disease level change. So we found we made a 3 type of mutation on different places of the gene. Actually, we found that only knockout in the NLS side or before that side show kind of disease increase. But after that, the disease not change. So because when we when we clone that gene, so we have 3 different reports. One report for the like candidate as a PFT, another 2 when we our report believe that the FHB1 due to the knockout the susceptible gene, but another group they think because gain new function of resistant gene. But based on in the last several years, some work report, the more recent report in the Nature Genetics, they just use mutagenesis. They show that PFT, you knock out PFT didn't change the disease. So they can confirm PFT right now is not like FHB1. But another group, they use gene editing of different kind of commercial variety, try to knock out the HRC, try to develop a resistant line. They found that like I think 3 varieties, there are 2 of that knock out the HRC. They show the resistant. Actually, they support the loss function of HRC to confer FHB1 resistant. So like it's not like a new function right now. Pretty much they can confirm the work of what we reported before. But we found that the HRC not only kind of control the weak scab, it also can confer resistance to the rice blast resistance. So this just show evidence of the knockout gene. They also show a lot of function to confer resistance. So this is a white type. When knock out, the disease become smaller and the region size becomes shorter and narrower. So this overall disease level kind of reduce. So fungal biomass kind of lower. So also resistant to the rice blast. So we also conduct the try to conduct experiment to identify interacting protein with HRC. So because it's a 2 hybrid screening, we find like more than 10 different interacting protein. We pick up the one called CAXIP4 protein because this because the gene HRC have something to do with the costume binding, but we couldn't find any costume binding site in the inside of the gene. We thought might be something else help to interact to finding the COSM signal system. So we found that use this gene. We found that this gene have interaction with the CAX1 gene, so here. But when we add the HRC, so then the interaction is gone. So that the HRC interfere the interaction between the CXIP4 and the CX1. So then based on that fact, we propose a kind of one of interaction, the pathway is kind of thing, because in the COSM, so when without the HRC, the CSIP4 interact with CX1, so enhance the COSM transport. So then when HRC combined with CAXIP4, so this, because this suppress the interaction between the CAXIP4 and CAX1, so block the COSM transport channel, so that make a plant because become more susceptible. So, but there are still several other kind of pathway interact with the HRC to control the FHB resistance. So we are actually, for the breeding side, we try to replace FHB1 in a different kind of background. So this show that just one line we select from overland. You see this in field. This shows like pretty high resistance in field, the same field, the two different rows. So this one, like overland original plant. So in our disease nursery, they show pretty high disease level. So look like some plant when you select even increase use just FHB1, you can select very high resistant variety. So more recently, in last several years, we tried to pyramiding different gene. First we started with FHB1, FHB5, and some resistant gene on 5AL. And FHB9. So then we select some line. So this is in the scab nursery. So this line have at least like 2 or 3 genes. So they show all very high resistance here. But those lines kind of exported that control you see completely dead the head. So that's show that even you have like 2 or 3 genes that show pretty good resistance in the field. So this right now, so then we use this 3 gene because those plants we have trouble with the hard groom because of one from 5AL. So a lot of resistance will have hard groom. So we try to put the FB9, 7, and 1 in same line. Right now we have like put all of that in 15 hard winter with genotype. So those from like 5 or 6 states. So right now, most of the line we have already done, we send to a breeding program, let them to select in field for the other agronomic trait to develop a variety directly. So in short summary here, for FHB1 resistant mainly due to like loss of function mutation of HRC, and transfer FHB1 from SUMES3 source can significantly increase the resistance. But here, what I say is SUMES3 source, if you use a marker, so that definitely will work. If some other source, I'm not sure if not detected. So the resistance level in FHB1 carrying lines vary with the background, depending on how many monogene or susceptibility gene in the background. So FHB1 shows like additive effect with most of other resistant QTL. However, in some of Chinese landrace, some like FHB1 is have same sequence, but they may even have like a much smaller effect, or some you hardly detect an effect in that region. So that's why I can emphasize you have to, we will show the one from SUMES3 that definitely If you in breeding, you transfer that from Smith 3, definitely you will be able to increase the resistance. If from all the different kind of land race, we need to confirm that before we can use that in breeding. What I assume that probably have different FHB1 allele with a different level of resistance. So this needs kind of further research on that. Oh. So for the future work right now, what I'm thinking right now, we have like 3 major genes. So if we use this very well, if we put at least 2 with some kind of moderate susceptible background, we can create very high level resistance. So only thing is we just need to select one with a good agronomic trait that other variety to release. So we can use the several different methods. Either we can transfer FHB1 or FHB7 to like at least like moderate susceptible background. We can create kind of a MR and moderate resistant to resistant variety. Also, we can pyramiding 2 or 3 major genes in different background to create higher resistance through like a high throughput marker system selection method. So another way, use the new technology, we can knock out the susceptible gene, such as FHB1, to improve FHB resistance. But doesn't matter which way, still have a lot of variation, even you have a gene there, so that we still need to use effective phenotyping to select design the resistant level in field nursery or in greenhouse. So at multiple locations if possible. So phenotyping still needed, even we use the marker state selection to select final resistant variety. So, but the work we still need to do, like we need to discover, clone more major resistant gene if possible, and develop diagnostic marker for breeding to improve diversity of the resistant gene. So then, because we think there are a lot of susceptible gene there, so if we can develop an effective tool for quickly discovery of a susceptible gene, that we can create more target for the gene editing. So also meantime, we need to develop kind of gene editing pipeline for the breeding application, so that can use directly easily to use in breeding. So same time, we need to understand the resistance mechanism of a non-gene to manipulate them for the improving FHB resistance. So, and I think that's all for today's talk. We, as this working, a lot of people help from different universities and different department and USDA, and get especially like US GAP initiative give us supporting a lot, 20-some years. So I really appreciate that. So thank you. If you have any question, I'm ready for take some question.
Awesome. Thank you, Dr. Bai, for such an insightful talk. It's Lovit here. If anyone has any questions, it's the time to ask now. Maybe I can ask a quick question here about FHB susceptibility genes. For my doctorate, I worked on susceptible genes. You hear me well? Yeah. So what we see, like in order to have a highly resistant cultivar, is it like, can we mix all these susceptibility genes that we can discover through transcriptomics with these resistance gene to achieve higher resistance? Like, do you think those crossing, like having a knockout and having a resistance gene, gonna work in the future we see for Fusarium head blight? Or I don't know how we can use resistance and susceptibility together to improve the resistance.
Yeah, you can actually treat them as a kind of independent gene. For resistant gene, you can just transfer there. For the susceptible gene, if you confirm that's a susceptibility gene, you knock it out. So then you can make cross. So
okay.
yeah.
Hi, Guihua. Thank you very much for the presentation. I really enjoy it here. One question about the THRC, the FHB1. So this loss of function is on 3B. Did you check the loss of function for the homolog on 3A and 3D? Do they also show the resistance or there's no homologs?
Yeah. When we clone the gene in that specific background, that only like 3B, so that show the function order, because the problem is only this genes actually we find that overexpressed in susceptible variety. So, say clock or some susceptible variety, they're highly expressed. But
Okay.
in other two homolog and the resistant one, you can actually expression is very low.
I see. Okay.
So we cannot confirm because this loss function. We cannot confirm that A and D homolog also have resistance or not. Yeah, but we just change the D, they show the difference. But look like one paper I just mentioned that they did the gene editing with different variety. They found that like D genome homolog also have effect. So they still need kind of more work on it. So
Okay, I see. Yeah. Thank you very much.
yeah, you can check that paper, the just more recent published in Plant Biotechnology. That
Okay, I will. Thanks.
yeah.
I have a quick question on you showed like you tried to have FHB1 and FHB5 together and then FHB1, FHB7. I just wondering I might have missed it. Like when we put both those QTLs together, do we see any penalty on yield or anything like that, or everything looks fine?
A gene what?
Like when we put FHB1 to and FHB5 together, like definitely there is improved resistance, but is there any other penalty, like yield penalty or anything?
So actually for the FAP, what I say, we try to put the FAP5 and the one FAP5L, that one shows like higher resistance, but that one we got like everything else okay. This resistant, very high resistant, but groom too hard when we thrash it, a lot of harder separate that. Perhaps that will not grow in a real variety. But besides that, all the trait will be okay. We did like FHB7 right now because we don't work on the 5A anymore. A 7, a 1, and a 9. So look like the spike look normal. So we haven't implemented a yield test yet. Yeah.
Awesome. Yeah. Thank you.
Yeah.
Any other questions for Dr. Bai? Anything in the chat? No. If not, we can thank Dr. Bai for such an insightful and great presentation. And everyone have a great weekend.
Thank you.
Yeah, thank you so
everyone.
much.
Have a good weekend. Okay. Bye-bye.
Bye.
Thank you, Dr. Bai. Yeah.
Mm.